Hi everyone, my name is Ziheng Duan and I'm a third-year CS PhD student at UC My advisor is Professor Jin Zhang. Today I'm very excited to talk about my work called iHerd, which was published in PLOS Computational Biology. And iHerd is a tool that helps us understand and identify kin genes in disease by analyzing how genes networks change. Okay, before we dive into the details of iHerd, I would like to introduce some background about gene networks. Okay, let's first look at the gene regularity networks, or GRNs. Imagine that there are three genes X, Y, and Z. In healthy people, X regulates Y, and then Y impacts Z. But for diseased people, it's a bit different. Well, for this simple network, we can easily tell the difference. In control, gene Z is regulated by one, but in disease state, it may be regulated directly by gene X. To illustrate this further, let's consider the dynamics within the gene co-expression networks, or GCNs. We can observe that the connection pattern between gene X and Z undergoes a significant alternation when comparing the healthy individuals with the disease. So it is these pivotal changes that our research targets and aim to understand. However, in the real world, it's not that simple because gene networks are huge. Take a look at this slide. We have a gene network with almost 7,000 genes. It's really tough for us to directly spot the difference between this network with another gene network also with 7,000 genes. So that's why we need a smart system to help us clearly show how this network change. However, detecting these changes is just the beginning. The real challenge is the quantification. Consider gene 1 and gene 2 in this diagram. And both genes show changes from control to disease. These changes are measured by the distance in the latent space. It's essentially how far those genes are shaped within their network from control to disease. Notice the difference in the distance for gene 1 and gene 2 between healthy and disease. Gene 1's cluster is more displaced than gene 2's, suggesting that a significant alternation, potentially even a pathway change for G1. Understanding the scale and the biological implications for these changes is what our research is diving into. We cannot just identify changes, but can interpret their biological impact. So this is where our iHerd framework comes in. Just check out this framework. We start with a big complex graph at the top left corner. And we use a graph Gaussian model to cluster its similar nodes together and guide a Gaussian graph with fewer nodes. Next, a graph learning method is applied to the Gaussian graph and guides the embedding for each node in the Gaussian graph. Later, we design an embedding refinement model to generate the final embedding of nodes in the original complex graph. And then we use a hierarchical embedding alignment to align the embedding of different networks and the different resolutions to quantify the network changes. Thanks to the hierarchical framework, iHerd simulates the gene pathway and can capture the different levels of information to better detect network changes under different resolutions. Before we dive into our findings, let's quickly define TFGRNs. Transcript factor gene regularity networks are complex systems where transcript factors interact to regulate gene expression. Now focusing our results, we see a distinct hierarchy in this network with two cell lines, K562 and GM12878 cells. The upper level of this hierarchy represents the top tier TF, which are crucial as they indicate the difference in gene expression between healthy and tumor cells. On the other hand, the bottom tier TF, although lower in the hierarchy, they are involved in numerous binding interactions, underscore their neurons but the substantial roles in gene regulation. This hierarchy is vital for understanding how genes are controlled in varying cellular states. Let's look at the experiment part. First, we analyzing the hierarchy change within two transcription factor GRNs. After we get the embedding for each TF, we plot in the UMAP. If you see finger A and C, they show three distinct groups. But there is the interesting part. When we check the ratio of in and out degree for each group, we notice that each group was, for example, the red group, the ratio is very low, almost equal to 0, which means there is no in degree, but a lot of out degree. Another group, or the green group, the ratio is equal to 1, which means this group is mainly about passing message. And the blue group, their ratio is very huge, and this group is mainly following orders. It's like finding commander, message, and soldier patterns in GRNs. Next, after we did the alignment from finger E to F, we can directly compare the network changes. Finger H to J also shows top 3 TFs with the largest distance in binding space, and all of them change their roles from the soldier group in control to commander group in disease. So in order to prove the superiority of iHerd, we also designed a simulated GRN experiment. Since it's hard for us to get the ground truth, we manipulate the true GRN and generate a simulated GRN, and then put those two GRNs to different methods to quantify network changes. Let's focus on finger A for this finger for a second. On the top left, you can see how gene BCIVF1 connects with others. And on the top right, how it looks in disease. We mimic the disease pattern and change the BCIVF in the healthy network to create our simulated one. The key here is the gene BCIVF1. It should show the biggest difference between the real one and the simulated one. And we can use the recall rate to measure the performance of different methods. Here is some baseline summary. If we only use the no degree to quantify network changes, when the in and out degree remain the same, we cannot prioritize the red one. The same for clustering coefficient. And if we use the page rank, a small changes will cause all changes for each node due to its global scope, so it also struggles. For contrastive learning, if the change node remains the same clustering membership, there is no way to detect it. So the recall rate in table 2 also proves the effectiveness of our method. Next, we move towards another network called gene co-expression network, or GCN. We collect the single-cell RNA sequence data from human prefrontal cortex. And first, we did a cross-cell type or neuron with non-neuron analysis. For example, iHerd detect 2 genes, PPARG and RUNX2. All of them are differentially expressed genes. And iHerd can also highlight some cell type specific divergent genes in brain disorder. Here are 4 examples. To prove the effectiveness, we also did a simulated GCN experiment. Here we mainly focus on the LDG and the EDG classification task. LDG means late divergent gene, which shows some changes from control to disease, but it does not change the pathway. EDG means early divergent gene, which shows changes from control to disease, and it also changes the pathway. So we also did some simulation here, and table 4 shows the result. Due to the hierarchy design, iHerd can effectively learn the within cluster and the cross cluster pattern, thus achieving the best performance in this LDG and EDG classification task. Next, here is some robustness and biological analysis for iHerd. First, the distribution of the community site for each run and the overlap between communities, between runs. And the goal term enrichment analysis show that the community or clusters generated by our method are stable and biologically meaningful. So after a lot of analysis and experiments, we can draw the conclusion that iHerd can faithfully highlight the changed TFs and the divergent genes across different cell types and conditions. And with the exponential growth of disease data, iHerd can be a useful tool in this field. Finally, I would like to express my sincere gratitude for the members of the SCORE group for their contributions. The Jones Lab team for their expansional work and dedication is given special recognition. I particularly appreciate my advisor, Dr. Jing Zhang, for her invaluable guidance and support. We are also thankful for the support from NIH, whose contribution has been essential for our research. Okay, thanks for listening. Any questions?
Thank you. We have time for a couple questions. So I have one. So for iHerd, is it still in development, or is it something that people can use?
Sure. We have published iHerd as an open source software, and it's free to download.
Thanks.
And you can check our Plot CV paper, and it will give you the link to download. Thank you.
Okay. Yep. So we have one in the chat. So asking, how does iHerd work on primary patient samples such as CD4 T cells?
Sure. For some specific network, I think first we need to build the graph. Let's say, for example, we have the control sample, and we can build the regularity network for control sample, and then we build another sample for disease. And after running our method, we can tell you which gene changed the most, and we can give a ranked list.